FLEXcyte 96 plates are designed and provided by InnoVitro for high-throughput cardiac contractility measurements using the FLEXcyte 96 benchtop device, an add-on system for the CardioXcyte 96, which is provided by Nanine Technologies. Upon arrival, open the parcel carefully. You will find a printed FLEXcyte 96 plate handling guide, which should be read carefully before you start your experiment. You will also find for each FLEXcyte 96 plate an additional lid to be used during your experiment, and store the FLEXcyte 96 plates, which are covered in sterile and vacuum-sealed plastic, in a dry and dark place at room temperature. To avoid contamination before use, transport the sterile and vacuum-sealed FLEXcyte 96 plate, as well as the additionally supplied lid, into a sterile environment like a laminar flow hood. Place both items on a flat surface and remove the vacuum-sealed packaging carefully. To enable the FLEXcyte 96 plate flexibility, the bottom of each well is covered with a very thin membrane, which may be damaged if touched from inside or outside. Hence, the FLEXcyte 96 plate is protected from above with a regular 96 plate lid and from below with a blue membrane protector. Since the blue membrane protector is non-transparent, make sure to exchange it with the additional regular 96 plate lid provided. In order to do this, you have to break the banderole that is wrapped around the FLEXcyte 96 plate. Now lift the blade together with the upper lid carefully while holding the membrane protector down at the remains of the banderole. Place the FLEXcyte 96 plate upon the additional lid by stacking the plate onto the lid. Do not turn the lid upside down to avoid disruption of the membranes. Keep the FLEXcyte 96 plate stack onto the lid until measurements are performed in the FLEXcyte 96 system. Before you start the seeding procedure of your desired cell type, a fibronectin coating of the membranes is necessary to ensure optimal cell adhesion. Prepare a total volume of 11 ml fibronectin coating solution with a concentration of 10 microgram of fibronectin per milliliter. Transfer the coating solution into a sterile reagent reservoir. Leave the FLEXcyte 96 plate on a flat surface and hold the 12-channel pipette in a 45-degree angle with the tips against the inside wall of each well to avoid direct stress for the membranes. Now slowly dispense 100 microliter fibronectin coating solution into each well. As an alternative to manual pipetting, we recommend using the Integra Assist Pipette robot. Please refer to InnoVitro's plate handling guide for recommendations before use. Don't forget to inspect that all wells are evenly covered with coating solution before you transfer the FLEXcyte 96 plate back into the incubator for at least 3 hours at 37 degrees temperature. After 3 hours of fibronectin incubation, start the preparation of your desired cell type. Regarding human iPSC-derived cardiomyocytes, prepare the cells according to manufacturer's guidelines and adjust the cell number using InnoVitro's plate handling guide for recommendations regarding cell densities. Once you have prepared the cell suspension, take the FLEXcyte 96 plate out of the incubator and place it onto a flat surface in the laminar flow hood. To aspirate the fibronectin, use a single-channel vacuum aspiration system. Hold the FLEXcyte 96 plate with one hand in a 45-degree angle and carefully aspirate the fibronectin with the other hand by starting at the bottom rim of each well where the fibronectin accumulates. Be careful not to disrupt the membranes with the vacuum aspiration pipette during the aspiration procedure. Immediately after removal of the fibronectin coating solution, transferred the cell suspension into a sterile reagent reservoir. You need a total volume of 11 milliliters to seed the entire FLEXcyte 96 plate. Add the cell suspension using the 12-channel pipette. To ensure the protection of the membranes, leave the FLEXcyte 96 plate on the flat surface and hold the 12-channel pipette in a 45-degree angle with the tips against the inside wall of the well before slowly dispensing the cell suspension. After all desired membranes are seeded with your desired cell type, transfer the FLEXcyte 96 plate into the incubator and let the cells settle overnight. Perform a complete culture medium exchange the day after seeding. To ensure that the cells attach to the membranes successfully, inspect the cells under the microscope before you start the medium exchange. However, you need to remove the bottom lid of the FLEXcyte 96 plate carefully before you look through the microscope. When you are done with the cell examination, stack the FLEXcyte 96 plate back onto the bottom lid and proceed with the medium exchange in a laminar flow hood. Transfer the freshened pre-warmed culture medium into a sterile reagent reservoir. Use 22 milliliters of freshened pre-warmed medium from your desired cell supplier for the entire FLEXcyte 96 plate. Do not remove the medium of the entire plate at once to avoid drying of the cells. Instead, aspirate 4 rows at a time using the single-channel vacuum aspiration system and add the fresh medium immediately afterwards to the 4 aspirated rows. Hold the FLEXcyte 96 plate with one hand in a 45-degree angle and aspirate the medium with the other hand by starting at the bottom rim of each well where the medium accumulates. For adding fresh medium, leave the FLEXcyte 96 plate on a flat surface and dispense 200 microliters of the medium into each well by holding the pipette in a 45-degree angle against the inner wall of the well to avoid direct stress for the cell layer. Repeat this step for the next 4 rows to complete the medium exchange for the entire plate. A medium exchange is recommended every other day, apart from weekends, until you are ready to start your experiment. After 3 to 4 days in culture, the cardiomyocytes form a regular beating syncytium, which can be seen easily with the naked eye. Each well should now contain a final volume of 200 microliters. When you are ready to start your experiment, make sure to perform a final medium exchange 4 to 6 hours before compound addition to ensure an optimal nutrition level for the cells. Have your measurement plan ready and prepare your compound working solution using a regular sterile 96 deep well plate. For example, prepare a 4 times concentrated working solution, transfer the compound working solution into the incubator for at least 1 hour to adjust it to the same conditions of the FLEXcyte 96 plate. 1 hour before you add the compounds to the cells, remove the bottom lid of the FLEXcyte 96 plate and transfer the plate into the FLEXcyte 96 benchtop system. Do not forget to perform a baseline measurement of the FLEXcyte 96 plate before you add the compounds to the cells. In order to do this, perform 3 sweeps for a good reference baseline. Perform the compound addition directly within the FLEXcyte 96 system. Lift the upper lid and carefully remove 50 microliters of the medium in each well. Immediately afterwards, add 50 microliters of the compound working solution and put the lid back on. Try to perform the step as quickly as possible and start your measurements right after you added the compounds. The number and intervals of measurements may differ depending on whether you want to do acute or chronic compound analysis. For recommendations regarding specific protocols for commercial human iPSC-derived cardiomyocytes, please refer to Innovitro's plate handling guide or contact Innovitro or Nanine Technologies directly. For questions regarding the FLEXcyte 96 benchtop device, please contact support.cellular.networks@nanine.de. For information regarding cell handling or FLEXcyte 96 plates, please contact info@innovitro.de.